Thanks everybody for joining the May 2024 monthly MIDAS webinar. Today we are very pleased to be able to welcome Nguyen to give a talk to us. Han is one of the folks who I consider to be really the unsung heroes of U.S. science and our national science endeavors. Han is a program officer and program director at the Division of Biophysics, Biomedical Technology, and Computational Biosciences of the NIGMS, the National Institute of General Medical Sciences. And as a program officer, Han is one of these people who helps shepherd along the work that we do and helps bring things from proposal through review and to funding. And he's really among the large cadre of folks who work with NIH to help this process and make sure that the good science gets funded and that the work moves forward, and to make sure that the money from the federal government is being spent well in science. And I know it's really not an overstatement to say that those of us who work in university-funded research couldn't do our work without Han and a lot of people like him who get very little credit and very little recognition. So it's always my pleasure to give that where I can. Han does a lot of work in a broad range of things involving theoretical computational physics-based methods and as applied to computing, modeling theory, anywhere from atoms and molecules to work like we do at MIDAS. And Han's got a strong background that he brings to this, including a PhD in chemical and biomolecular engineering and postdoctoral work in biophysics. And so I encourage folks, if you're writing grants, to get to know your program officers, talk to people like Han. I know they're always available to try to help folks when they come to the question of, hey, where do I send my proposal and what's the best way to try to get funded for these ideas, things like that. That's what the program officers are really, really great at. And so today, Han is going to tell us about some of the processes at NIH and the opportunities specifically at NIGMS. And so this is all something that anybody who's going to do any real work in science should be thinking about and aware of. And so I'm going to hand it over to Han and encourage folks to think of your questions and bring them at the end of the talk so we can use Han's time well and really pick his brains to understand how to work this process. So with that, Han, thank you, and I'll leave it to you.
Thank you, Harry, for that introduction. I am glad to be here to introduce some information on the funding process at NIH and funding opportunities at the National Institute of General Medical Science, or NIGMS. As you know, the mission, the mission of NIH is to provide support for medical and behavioral research. And they try to support the kind of research that can seek fundamental knowledge about the nature and behavior of living system and applying that knowledge to enhance health, lengthen life, and reduce illness and disability. NIH is a very complex system. It has 27 institutes and centers. We refer to them as ICs. And NIGMS is one of those 27 institutes, and it's the fifth largest IC in terms of the budget that we receive from the Congress. So 80% of our budget is devoted to extramural grants, contract, and cooperative agreements. Because there are 27 different IC institute, and each institute have their own mission, so we have to be very careful in accepting application to our institute. Our mission is to support basic research that can increase the understanding of basic biological processes laying the foundation for future advances in disease diagnosis, treatment, and prevention. We fund research on how living system works at a range of levels, from molecules to cells to tissues and organs in research organisms, human and populations. One thing that we do not support is that we don't support research that focus on a single class of cell, tissues, organ, and disease, unless they are used as model for elucidating basic principle. The reason why we don't do that, because there are 20 other IC that fund those kind of work. We just fund the basic research that are generalizable to a range of different cell, tissues, and organ diseases. So not just a single one of them. And in addition to those kind of research,
Can I
yes,
interrupt for a second?
yes.
You are in you're showing us not the you're showing us the presenter note mode. So if you can switch to show, yes.
Oh, sorry
that
about that.
off. Try that's better. Thank you.
Oh, yes. Yes, so in addition to the research, basic research, we also support technology development as well. So technology development, I will mention, discuss in more detail later. So as long as you can develop a new technology that can allow all the researcher to perform research, understanding general principle, mechanisms, and processes, then we accept those kinds of application as well. So we understand that the funding landscape at NIH is very, very complicated to navigate. So therefore, we encourage everyone to contact program officers for help. We can provide all kind of feedback. So here I can discuss a few things that we can do. We can provide feedback on suitability of your proposed work for a particular institute's mission. For example, for us, we can tell you whether your proposal is suitable for our mission or not. And we can tell you whether it's appropriate for a certain funding opportunity. It's called NOFO. Then if you have question about the requirement for your proposal, we can clarify programmatic requirement and administrative policy as well. And in some cases, if we can have a more understanding of your proposal, we can provide general tips and strategy on writing strong application and give you provide you with resources that may help you in preparation for strong application. So those are the things that we can do, but what we cannot do is that we play a co-author for your application. We cannot edit any part of your proposal at all. What you can do, you can send us an abstract and we can look over that, but we cannot provide suggestion how to modify your proposal to provide changes to your research plan or your experimental design. And of course, we cannot share any inside information. All the information have to be in the public domain already. Yeah, so for this reason, I encourage you to contact your program officer. So how do you find a program officer at NIH? We have resources that can allow you to find potential program officers and institutes. So we use this, you can use this database called report. All you have to search for is NIH report, and it will show you this page. and then click on Matchmaker so that you can paste your specific aims or your abstract into the required space, and then it will provide you with a similar proposal that have been funded at different institute. And you can click on the program tab, program officers tab, so that you can see the email, contact information, so you can contact those program officers. So the other task that you should do is to find appropriate grant mechanisms. There are many different funding mechanisms available for you. So depending on what kind of level of training and independence you are, we can provide you with a funding opportunity as a fellowship or for training or for career development or for research. If you are at a particular institute that only support research performed by undergraduate student, we have a particular mechanism as well. That's of-16. If you have mostly student of underrepresented population in biomedical research, we have a mechanism called R16. I will go over that in a little bit. And depending on how well developed your research problem is, if you have if you add a exploratory stage in the beginning, you have an idea, but you need to explore it further, then you can apply R21 without prelim data. Or if you have strong prelim data already, and you can apply for a full R01 grant. If you want to fund your whole research program, the majority of your research program, we have a mechanism called MIRA. So in this case, we fund multiple projects rather than one specific project. So let me summarize that in this particular slide at a different career stage in you can apply for different mechanisms. So as a graduate student, you can apply for a pre-doctoral individual fellowship. So that's F30, the one in yellow, the training grant. You can apply it with your institute because we provide funding directly to the institute or your department. So therefore, you can get that training grant fellowship from them. But the one, the F30, you have to apply directly as an individual. Similarly, as a postdoc, you can apply for a fellowship to support your postdoctoral work as well. And in between, we have transition grant. If you are a graduate student and you would like to transition into a postdoc, we have a award called F99K00. And similarly, we will support the development of a postdoc became a professor, independent researcher as well. So this one is called NIH Pathway to Independence Award, or K99R00. And as an independent, once you become professor, as an independent researcher, you can apply for a range of different award right there. So I will spend majority of the time over these different mechanisms. So the first one is the individual fellowship for a graduate student. So in this case, NIGMS would support your PhD thesis as long as your project fall within our mission. And in this case, we require a research training is individualized and tailored to the candidate strength and the opportunity requirement. We can provide support to for 3 years. So here I provide a link, and I'd be happy to share the slides so that you can have a link to individual funding opportunities. So as I mentioned earlier, we also have a developmental plan to support graduate student that became postdoc. So therefore we have this F99K00 grant as well. Once you became a postdoc, you can apply for F32. So this one require a mentorship with your advisor. You can find funding to support your own postdoctoral training experience, and the research project must be integrated with the training plan. So we can provide you 3 years of support. So the pathway to independence is one of the very prestigious award that you can get. So it's K99R00. It provides 5 years of support, consists of 2 phases. K99 is for a postdoctoral researcher, and then once you become an independent researcher as a professor, then you can receive funding through the R00 phase. So if you have more information, if you need more information, here's a contact person. You can make inquiries. In addition to that, we also have the Mosaic program that is to promote the faculty diversity as well. So if you qualify for diversity, you can apply to the Mosaic program. Is very similar to the K99R00, but it's specialized for faculty diversity. But I want to mention a few more things about the K99R00 award. The eligibility is that you cannot have more than 4 years of postdoctoral research experience already. And because this award requires mentorship, so therefore you have to be in a mentor postdoctoral training position when you apply. So the individual that are not eligible is the one that have held an independent research faculty or tenure track faculty position already. Those applicants are not qualified to apply. And if you have received some kind of independent research grants already, you're not qualified. But one thing that I want to mention is that this is the only one funding opportunity that requires no U.S. citizenship. What this means is that anyone can be a U.S. citizen or a foreign postdoc can apply. And so there are 2 phases right here, the K99 and the R00. For the K99, when you first apply, the applicant's institution is responsible for determining and documenting that your visa allow you to remain in the US long enough to complete the K99 phase of award. And then when you became a faculty somewhere, the new institution will be responsible to determine and document that your visa allow you to remain in the US for this award. So hopefully by this time, your new institution will help you to obtain a permanent resident visa so that you can become a US citizen. So this award is very important for foreign students and domestic students. So now, once you became a faculty member, you can apply for independent research award. So R15 is when you end up at an institution that have received very limited NIH research support in the past 7 years. That what I mean is that that institution received less than $6 million total in 4 of the 7 years. When you can qualify to apply for R15 to support small-scale research projects, where majority of the researchers are undergraduate students. So in this case, you can receive 3 years of support in the amount of $375K direct cost for the whole period. So here I listed the contacts so that you can make inquiries. If you are at an institution where it received a limited amount of NIH funding in the past 7 years, and it serves students from groups of underrepresented population in biomedical research, then you can apply for an award that lasts for 4 years. It can be in the amount of $125,000 direct cost per year if that's the first time applying. For experienced faculty investigator, you get $100K per year. Okay, so let's say that you are at a institution, a research heavy institution, and you can apply for R00 to support your research group, and the parent R1 is the most popular one. All institute participate in this funding mechanisms as long you can provide a discrete project with specific aims, and the research have to fit a certain mission, then you can qualify to apply. So this is a very broad mechanism. You can go on NIH report database to find appropriate program officer, contact them so that you can make inquiries about this funding mechanisms. At NIGMS, we have a particular mechanisms called MIRA, is Maximizing Investigator Research Award. It can provide 5 years of support, so therefore it provides you a lot of flexibility, stability, and also it support a research program rather than a research project. So therefore, it doesn't require specific aims. What that means is that you can talk about your research program at high level. You don't have to go into the specific aims for your experimental design. So along the way, it provides you flexibility if you want to change the direction of your research. When you make a new discovery and you want to pursue that line of research, you can do that. As long as that direction is within our mission, we allow investigator to do that. So here the only requirement is that we a single PI have to devote at least 51% of your research effort to on the MIR award. So we have 2 different NOFO. One is for early stage investigator, those who have obtained their terminal research training, for example, PhD in the last 10 years, but have not received any major substantial award with us yet. Then you can apply. So in this case, you don't need to have prelim data, or you don't need to have independent publication. You can just apply directly in as long as you qualify as a early stage investigator. If your PhD already passed 10 years, but you haven't received any funding from NIH, you qualify as a new investigator. You can also apply to this NOFO called 22180. And there's no requirement for you to apply. You don't need to have any major funding. You don't have it anyway, so you can apply. But for the established PI, those have consistently received funding from NIH, then you need to have one single PI or one from us first. And then we would convert that into a mirror when your mirror application is approved. So, so far I have mentioned all the different funding mechanisms that allow you to perform research. But what if you develop a new technology and your technology can be used for many people, for many different projects in different settings? You can apply through our technology development programs as well. So as I mentioned earlier, that you can apply as all-1 or all-35 if you're addressing biological questions using established technology already, but you're developing technology at the early stage where you have to provide proof of concept and value. You can apply through the all 21 mechanisms for the technology development grant. And if your technology is more mature and now you need to provide prototype validation, so therefore you can apply for an all-1 award for the technology development focus grant. So I should mention that the mirror will allow you to incorporate technology development in the proposal as well. So the R35 can have two components. It could have one project addressing biological questions and one project on technology development. That's okay. And if your technology can be can have potential for commercialization, we can allow you to apply if you have a small business. So in this case, you can apply through the STTR. That's a small business technology transfer, or the small business innovation research. So SBIR. The difference is that for the technology transfer, you have to partner up with a institution. As a small business, you have to partner up with an institution where you transfer the technology from that institution to your company. But for the SBIR, you can develop your own technology as a small business goal. So here we have contact information that you can make inquiries. So as I mentioned earlier, that I belong to the division of biophysics and biotechnology and computational biology. But the branch I belong to is called bioinformatics and computational biology branch. So for this branch, support bioinformatics and computational approach that can join biology with computer science, engineering, mathematics, biostatistics, and physics, so that it can provide new technology and for a broad application and uses. So we fund, here are the many topics that we fund for modeling and computational method development, for bioinformatic tools and analysis method, knowledge base and data repository, for biostatistics and quantitative analysis of biological data, for health informatics and infectious disease modeling. So I will talk a little bit more on infectious disease modeling because that's important for your community. But one thing I want to mention first is that we also have a JOY program with NSF. So this JOY program called Biomath is to support fundamental research in mathematics and statistics that address questions in biological and bio sciences. The requirements is that you have to have a biological problem that are complying with our mission, and you proposing to develop innovative mathematical statistical computational methods so that you can integrate the mathematical approaches to address important biomedical problems. There are two tracks that you can apply. One have a small amount of money, relatively speaking, for 600K for 3 years for a new team that working on a high risk, high reward projects. For track 2, you can get up to $1.2 million total for established teams that working on large scope projects. The motivation for this program is to allow people to apply directly. Let's say that you have been getting funding through the NSF, you can still submit your proposal to NSF, but when one is approved for funding, we can transfer your application through NIH system, and we will consider as a all 1 award. So if you're interested in this program, you can contact me for more information. So one thing that I want to mention more is on the infectious disease modeling. So we welcome any application that working on modeling of infectious disease. So for our mission, it has to be very general. So this modeling effort is to improve detection, control, and prevention of emerging infectious disease. So if you just work on a single disease, we won't accept that. So if you're working on a problem that is applicable to multiple diseases or new technology that can be generalizable to multiple diseases, we will accept that. Otherwise, your proposal will go to, let's say, NIAID or somewhere else. So the funding mechanisms that we encourage you to apply is the R15, R16, R01, R21, R35. Or if you want to apply directly to NSF, you can go through the Biomath program. So again, I just want to mention that for infectious disease, if you want to focus on a single disease, that application will not be accepted through our institute. It probably will be routed to a different institute. Yeah, so I want to spend a little bit time on early investigators because these are the ones that would need more guidance. And based on my own experience, I understand that the whole biomedical research is very complex, especially trying to fund funding for your own research group. So we, as program official, we are here to help you. So feel free to contact us. So as I mentioned, if you need mentorship, you can apply for the transition award. If you have no prelim data, you should apply for MIRA early in your career. And if you have prelim data already, and you have a specific project that you really want to get funded, you apply for R01 And one thing, as an early stage investigator, you have this privilege that you can apply for both mirror and all-on with overlaps at the same time. So therefore, it can maximize your chance of getting funding. For established PI, they're not allowed to do that. Or so as an ESI, I will encourage you to take advantage of this to apply both mirror and all one at the same time. So that concludes my presentation. So I'd be happy to answer any question that you may have, or send me email directly.
Great, thanks, Han. We just have one question here that somebody asked is, what are you willing to share the slides?
Yes, so I will send you the slide and you can distribute them to them.
Fantastic.
You don't mind? Yes.
We will send those out. Floor is open to anybody in the audience who might have some questions. Please either unmute yourself, go right ahead, or put something in the Q&A. Barring that, I'll throw out a couple of questions, Han. One of the things that I know I struggle with sometimes in looking at solicitations and figuring out where things go is the question of the relationship here between NIGMS and NIAID, and allergies and infectious diseases in terms of infectious disease modeling. Now,
Yeah.
you mentioned how if it's going to be a single organism, then it would have to go to NIAID. Do you see other differences between what the groups would fund and what else might guide somebody to go one versus the other?
Yeah, so if we look at the proposal that focus on a single for this case, let's just focus on the single disease only and looking at, let's say you just focus on flu right now, and you have particular question that you ask, and this kind of question we say, oh, this kind of question is too specific for flu only, then it should go to NIAID. But if you're asking question that say, oh, this question are very, very generalizable, it can be applied to flu and COVID at the same time. You're looking at the emergence of mutable disease at the same time, then we'd be happy to take that. So that's a yes, there's some overlap here, but the distinction is that it has to be generalizable to mutable diseases to the to many systems. Yeah, so that's our mission is for basic research only. It cannot be too clinical. It cannot be if it's too clinical, it will go to NIAID. Yeah. So
if I might just interpret what you're saying, it sounds to me that for somebody in infectious disease modeling who wanted to write a proposal and have it be of interest to NIGMS, that demonstrating their techniques or their methods on multiple organisms, multiple types of outbreaks might be a way to make that point more strong, to make it clear that you're not just focusing on one. Even if you're using, say, flu as an example, you might want to add on dengue as another example just to show the generalization.
Yes. So let's say that you're developing a new model for infectious disease, and you say that as a demonstration of the utility of your method of your modeling technique, you will apply it to dengue. But then we would ask you to provide additional example as well. We say, can you apply it to one more disease, so that therefore it demonstrates the utility of your method to a broader system, a broader system of infectious disease. So that's a distinction right there. that your technology, the question that you trying to answer is broad, applicable to many diseases, and not just specific to one.
Great, thank you. I think that's an important point that people should really keep in mind as they prepare proposals.
Yeah. So I should mention that you probably wonder why we pay so much attention to this, and from your own point, from your perspective, why do you care if you want your application to go to our institute or go to a different institute? Because if you're looking at infectious disease, your application will be accepted at NIH unless it's really our NIH mission. But anyway, so the reason why, maybe for your own perspective, is that from your own benefit, is that NIGMS have no pay line at all. We publish our funding record in public. If you go to this website, if you search for NIGMS in the loop, you will get updated information on how we fund a all-one or a mirror based on the score, and it can be very broad. It can be more generous than, let's say, NIAD or other institute. But on the downside is that we give you a smaller amount of money to start with compared to other institutes, because we want to fund as many groups as possible. So that's a funding philosophy that we have. So this kind of information is available online. You can search for that. So when I say that we have no pay line, what that means that we look at multiple factors. We look at whether you are early investigator, whether you working on something very interesting that can have a broad utility to the community, or you so there are many factors that we take into account. We don't just look at the review score and say that, oh, based on that review score, we should fund them. Anyway, yeah, so if you have any more questions on this, please let me know.
Well, we have another question from the audience here that I'll read, and then I know I can fire away with more questions if needed. But this question here, if one of the attendees asks, if someone has been co-PI on an EEID proposal transferred from NSF to NIGMS but had no other NIH funding, are they a new investigator or not?
Yeah, so the definition of early investigator is that you have to be the contact, the lead of that project. So if you're a co-I, you receive a relatively small amount of subcontract, a small amount of money to support, let's say, half a student or one student. We don't disqualify you from the early stage investigation status. You still keep that ESI status. So that's not a problem. So if you search for NIH ESI, you find more information on that. They give you a list of different award that may change your status or not, but being a co-I is not a problem.
Excellent. No, and there's a clarification question here asking, the new, not early.
Yes.
So the same thing applies?
Yes, new investigator, that means that your PhD is older than 10 years, but you haven't received any major funding from us. You may have received some, your name may be attached to a grant as a co-I, but you haven't received a major funding as a contact or lead PI, then you still qualify as a new investigator status. Yes.
Great, thank you. let me ask another question that I know always comes up when we are preparing grants. And I know this is something that, you know, I should back up and say, ask the program officer is rarely a wrong answer for many of these questions, right?
That's right.
But the question of which study section to request with your proposal, you know, when we send these things and we do indicate a study section that we think is best suited for it. And of course, the study sections cut across the institutes. Are there particular study sections that would be best or are well suited for infectious disease modeling proposals?
Yes. So depending on if you send us an application for the author-defined funding mechanism, we have a mere focus study session, and those study sessions are very broad. It can be yeah, it is for mathematical modeling. So that include a range of different systems, and infectious diseases included in there. So the Center of Scientific Review, which is a independent institute within NIH, it will the scientific review officer will look over your proposal and assign your proposal to the appropriate study session. So there's an online tool that you can use to figure out which one can be a potential study session for it. It's called ART, A-R-T. If you search for CSR, as in Center Scientific Review, and ART, it will come up as a database, and you can enter your abstract in there. It will tell you the potential appropriate study session. But to go back to your question, for infectious disease, we see if you apply as an all-one, usually it go to MAPS. You know, it's very general for NIGMS is and for other institute as well. It's for mathematical modeling. There are a few other ones that I go to. What I would recommend that when your proposal is assigned with a study panel, it will show up on your application status, and you can Google that. You can find out the reviewers on those study session, because those information are available to you. We have established a study session with long-term members who review it for 3 years or so. So therefore, those names of reviewers available. You can look them up. And if you look at the review panel and you use that database and you say, oh, the other panel is more appropriate, then you're welcome to contact the scientific review officer and explain to them. But they are the one that made a decision. The program officer have no control over that at all, because we operate independently. We have no control over who to assign for as a reviewer to proposal. The scientific review officer do their job independently. Anyway, that's a long answer to your question. But anyway, there's a few panel. I think MAPS is appropriate for infectious disease modeling.
No, that's great. Thank you. I know this is one of the things that I have always found to be a bit of a puzzle, and so it's a real trick to sometimes
Yes.
the way it would go. And I appreciate particularly your referring everybody to the ART tool, because that was one I wasn't aware of that one, so I'll have to use that in the future now.
Right.
Great, but it is a challenging thing to navigate for those of us who are writing the proposals. Okay, let me read another question here. If someone is part of an NIH, NIAID, TMRC application as co-PI, are they considered as co-PI even after they move to another institute when the project was sanctioned? So I think I'm going to have to ask the person there for clarification. Are you saying that you were part of the proposal as a co-PI, but then you moved somewhere else before the project started or before it was funded? Is that what you're suggesting? And I encourage the person who asked this question to unmute and clarify if they can. Okay, I'm not hearing any answer there. So,
Yeah, I can answer if that is a question, I can answer that one. Yes.
yeah, please.
So let's say you was in Pittsburgh right now and you got an award with us either as an independent, as a lead PI or as a co-I for the award, and you move to different institution, we will work with you to make that accommodation. You will receive the same amount of direct costs, and we understand that indirect costs, the overhead are different from one institution to another. We accommodate that as well. So that you can have the same direct cost amount. Yes, it's a complicated process. When you move to a different institution, we have to do more paperwork and all that. But the grants management, these are the people that work on your grants. One is funded already. This is the administrative staff that will work with you to make that transfer happen. That process may take a few months, but that's not a problem. So that require approval from us because we have to be able to make a judgment of whether the new institution is suitable for your research or not. So therefore, you have to provide information on the infrastructure, on the environment, on the equipment, the availability of equipment, and resources that you can use for your project. So there's a procedure for you to follow.
Yeah, and of course, as these things get into more specific and questions, okay, so the person here who asked that previous question is clarifying a little bit, saying that the co-PI's name was removed from the proposal after it was shortlisted. So the question there is this person then still eligible as an early investigator if they were removed from a proposal before it was funded? And it seemed,
yeah, that's very rare case. Yeah, if as a co-I, whether you on the proposal funded or not, you still qualify as an early investigator. Yeah, that's very specific. I don't have a complete answer for that. Perhaps you can send me an email. Yes.
yeah. So I think again, I appreciate the question, but this is where I also have to comment on, working with the NIH, that writing the grant is never the end of anything. Even if, if you are not funded, you're going to go back and you're going to talk to your program officer. If you are funded, you are likely through the course of the grant to have multiple interactions along the way with the funding officer and with the program, the grant administration staff. And there are often questions that come up that such as this one that we've just discussed a little bit, that don't have obvious answers.
Yes.
That require some discussion, and it's not infrequent that we have to ask the grants office, administration office at NIH something, and they will tell us, well, we're going to have to look into this and get back to you with an answer.
Right, right. Yes, that require multiple sources and all that. So
Exactly. And so there are, I'm sure, I have no idea of just how many edge cases there are, but there
right,
are probably a lot of them that come up. And
right.
so this is where I have to say that it's, you know, I said before about the wonderful work that Han and his colleagues as program officers do, but there's a whole teams of people behind the program officers who work on the post-award things and the pre-award on all the aspects of the submissions and on the funding and the paperwork and getting it all right. And I know when I get emails from all of these folks, any of these folks, it rises to the top of my list because they're the people who I want to make sure that I'm giving them what they need to do their jobs, that they can move things forward. Because often when I need them to move things forward, I like to make sure that they get the right answers. So we are just about at time here, and I'm sorry we can't go into more specifics of that question, which I do think is a pretty rare and non-trivial case. So I do encourage the person asking the question to get in touch with somebody in NIH to ask about that.
Yeah, so one thing I like to add to what you just said is that, yes, it's a very complicated process. And if you put in one application and it doesn't get reviewed positively and you don't get it funded right away, please don't give up. What we would like to see is that you come back with a stronger application, and we don't penalize you if you apply the third time, fourth time, even. We allow some mechanism allow you resubmission. Some don't, but you can come back as a new application. You can incorporate the critique offered by the reviewers and make your proposal stronger. So this is a learning process. So please don't give up. Some people, their feelings are hurt. They get frustrated. They don't apply again, and that's the last thing that we want to see. We want to see that you come back with a stronger proposal so that we can fund your research effort.
Yeah, I think that's a great way to end here since we're at the top of the hour.
Okay.
So thank you, Han, for your time. Very useful as always. Really appreciate it. And we will get the slides distributed as soon as Han sends them to us. And we will get the talk posted on YouTube as we usually do, hopefully sometime next week. And so thanks everybody for attending, and we'll look forward to seeing you at the June webinar.
Thank you. Thank you everyone for participating.
Take
Bye-bye.
care.